So I think, oops, I don't need an introduction right now. I hope after these few days together, we are quite know each other. And so first we planned a virtual visit to our lab, but due to technical limitations, we are not going to do that. So I just have some slides, and I brought together some examples of what type of research that we have been doing in photodynamic therapy and optical diagnostics. And it's really good to see that after Professor Svanberg, we try to have the same kind of a philosophy or strategy to try to, in our case, to solve one of or some of the medical problems that we have here in Brazil. And this I'm going to try to show you. And okay. So, in fact, the Biophotonics Lab, we are part of this Center for Optics and Photonics. This is supported by FAPESP, and we are just waiting for the new contract for this renewal for more 5 years, and we hope for more, and then 6 years, a total of 11 years. And all the infrastructure that we have so far was mainly supported by FAPESP, because we had the same CEPIRI program for the past 11 years. And in these centers, besides biophotonics, we have atomic physics and nanoplasmonics. And the CEPIRI is called for center that we must do research. So the name in Portuguese is pesquisa. We have to do also innovation and diffusion. And in this case, diffusion is for education in all levels. So we do have some programs for basic educations and also for training medical doctors, dentists, bringing these optical technologies. And also diffusion, FAPESP wants us to transfer the technologies developed to the private sector and also for the government sector. So in this case, we have to have these three important cores. Is not only research. This is one way that we can have some amount of the money that FAPESP is placing here, bringing back to the society. And so Vanderlei just asked me not to forget to mention we have a TV channel. This is a university TV channel that is all coordinated by our CEPID. And this is 24 hours of TV. So all the speakers that we had these past days, we have the lectures recorded, and will be in some of these days forecasting for this. But this is a local TV channel. And also we bring different speakers, different professors, even the graduate and undergraduate students to explain something related to science and to trying to motivate the young kids and the interest on science. So we do have some problems on motivating the students to go into physics and all the other science subjects. So this is the main topics that we have been working on the Biophotonic Lab at the present. So of course we are mainly going to be focusing today on PDT for cancer and infectious disease and optical diagnostics, because it's the main topics on these specific school. But we have been also working with different light parameters, illumination parameters that we can have, for instance, laser ablation, and understand how the irradiation parameters induce different changes and induce different response in the tissues. Another also that we have been working, it's low-level light therapy, that, for instance, we have this project that we are trying to improve the athlete's performance using infrared illumination during the physical exercise. So This is all ways of trying to have different metabolic and cellular effects just using the photons. So we start working with PDT back in patients in 1999. So the first patients that we treated here in Brazil, we used porphyrin injection. So it's a systemic porphyrin injection. So you have been from the past days already looking for this. So this is one case just to show this is a squamous cell carcinoma at the lip. And this is the result 20 years after PDT, so the cosmetic response is quite good. But of course, this patient must be away from the sun exposure for at least 4 weeks. And some other examples. So back that time, we just used the intravenous injections. All these patients were treated at the cancer hospital in Jaú, the place that we have also the other projects going on today. So this is like 3 months after this illumination. It was a single PDT session. And the patient was indicated for this case for PDT because he didn't, was not possible to give him anesthetics. So that's why it was, in this case, the option for treating with PDT. And this is 6 months later. So even though it's the second type of healing, we have also a very nice cosmetic response. Other anatomical region that is quite difficult to do surgery is this lower eyelid, because then the removal of this part of the tissue can really change all the functional of this area. And so this is the necrotic tissue that you are inducing with this kind of porphyrin systemic PDT. And this is another case, another example. For this, working on with we first started using these argon dye lasers. Of course, it's not something that's quite feasible for clinical practice, but in fact, this is in a hospital, cancer hospital in Jaú. So we always had the physicists together so they could align the laser, get the exact wavelength that we want with the enough irradiance, and then we could have this through these holes, send the fiber, so we could treat the patient on the other room. Of course, after that, we had this nice diode laser system that we could more portably take to different clinics. And these are some examples of some of the instrumentation that we delivered here in the physics department using LED system. And what's the good advantage of this system is because we can change the optical geometry depending on the anatomic site that you are going to deliver this light. So this equipment is, has even been licensed by the Brazilian agencies responsible for medical equipments. And, but specifically for Brazil, that we have a high number of non-melanoma skin cancer lesions. The systemic drug, because we have here a tropical country, so for these days we have been having an example of the nice sun that we have most part of the year. So in this case, the topical PDT is much more attractive, because then the patient can be treated and is not gonna be photosensitized for several weeks. But one of the limitations of these techniques, because we do have to have just a basal cell carcinoma, very superficial, up to 2 millimeters of thickness. And in this case, together with a company that is called MMA Optics, and in fact is one of the spin-off companies from our group, we designed this equipment that in fact we are having a dual PDT system, but because we can have the wide field fluorescence imaging, in this case, which is good, because we can check the protoporphyrin production after you place the cream, and you will wait for 3 hours, for instance. And depending on how it was prepared, or depend on the lesion characteristics, sometimes this production is not homogeneous. So it's a good system for the doctor that he or she can check just before, during the illumination, because something gets wrong and that there is no protoporphyrin production. Of course, there is no reason for illuminating for PDT if the photosensitizer hasn't been produced. So this is dual system. We have both probes, one for the fluorescence visor, and the other one is for illumination for PDT. And both probes are LED-based system. So this is just one example. So this is one BCC lesion, and this is 3 hours after the aminolevulinic acid, the ester form, was placed and with occlusive dressing. So you can check here this red, strong protoporphyrin production. And this is just after the illumination period with the red LED, which also you have been already seeing in other talks, that it can be a direct measurement of the PDT response, because if we had the red illumination and we check again, and if there is the same amount of protoporphyrin emission, probably the response is not going to really take place, or what was not going to take place in that region. So it's also one type of a nice clinical measurement that you can do in situ and during the PDT treatment of your patient. So this is some of the clinical results that we have 30 days after the photodynamic therapy. In this protocol, we have been using 2 PDT sessions, 1 week interval between those sessions. And this project has been funded by the Brazilian Bank for Social Development. And the reason is because we have both the equipment and also the drug that is also a Brazilian, is produced by a Brazilian company. So, and also the idea is to have these up to 100 centers having the same PDT protocol, and to have, after 3 or 4 years of this clinical trial, multi-center clinical trial, the response of the effectiveness of this type of treatment with this protocol. So at the present, we have already 65 centers trained in Brazil. So a team from our group that is formed by physicists, medical doctor, pharmacists, all are our graduate students. So they have their own PhD projects, but some of them took also part in this cancer program. And they go to the specific hospital or specific clinic, and these in all country, and they train the medical doctors, the nurses, with this specific technique. And so up to present, we have 65 sites using the same protocol. And now I'm just going to present one example of a case that I hope can show how we have been trying to develop this research in the lab, and how we have this, our philosophy to do all this type of experiment. So this is not a true fungus, but the life cycle is pretty similar. And so pitch insidioso induce skin infection that is called pitiosis. But the problem is because it's a fast growing infection, and it can go to your to the lymphatic system, and goes then for the whole body. So it's a life-threatening disease, and it can affect animals, like horses, dogs, even human beings. And the problem is because since it is not a true fungus, in one of the characteristics, because this microorganism doesn't have ergosterol in their cell wall, which is the main biomolecule that is targeted by antifungal drugs. So the life cycle of these P2 insidiosos, so in fact they colonize plants, and they are more present in the areas near rivers or in flooded regions. So most of the horses are contaminated when they live in flooded regions. And into the water, they start to produce the zoospores. And if there is any discontinuity on the skin of this animal, these microorganisms infect this area. And it can go to this type of very nasty skin lesion. And the problem of that is because the possible and the available conventional treatment do not have a nice response. As I told you, the antifungal drugs, they don't have the biomolecule that is the target of this main drug. So the main conventional treatment is surgical resection. But the problem is they have this growth like a fungus, so they have this hypha growth. So it's hard to know exactly until where they are going, and this is very fast growing rate. So in this case, first we just see the PDT effect in vitro, so looking at the microorganism, testing different photosensitizer, looking at the specific subcellular concentration of the photosensitizers, and later we do the irradiation and compare different protocols. At least for this case, we have to first see if it would have any response. So after checking in the in vitro experiments, we got into an animal model that is in rabbits. So in fact, the zoospores are injected, but as you can see, oops, not in here, but this is just the case with 21 of these animals. So we did with chlorine and porphyrin intravenous injections, because topical application with ALAL, we didn't have a nice response. These microorganisms do not produce too much porphyrin 9. And this is the type of deletion that we can use with this animal model. As you can see, it's not like the horse that is ulcerated lesion. But anyway, this is the only model that we have for this kind of infectious disease. This is just after photodynamic therapy, 48 hours. So we had already a big reduction of this mass. And this is 10 days after. So this is an idea what we always try to do with our PDT experiments. So in vitro, animal model response, and then we just finalize this study with 5 naturally infected horses. So in fact, these horses had been already took some surgery, resection, and this is in fact the second PDT section. Of course, in a horse, we are not going to use systemically injection of porphyrin or chlorine, because they are such big animals that we could have so much photosensitizer. So in this case, we just injected locally, and this is after the second PDT session. So in some parts, we even had some reepithelialization of these infected ulcer. And this is Lila, which is it was her master thesis study. And in those cars, we took a diode laser to do all this illumination. So one type of these lesion took us like 5 to 6 hours to illuminate this whole ulcer. So back to the lab, and then we just designed these LED systems. So now the next horse that we could be treating, this is one of the cases that we have the prototype design here, as Professor Svanberg was mentioning. We have this lab with 8 engineers that we can go talk to them and think on the design of the better properties of your illumination system, specific for each type of application. And these we have tried to do with all those different applications, PDT applications. So this is another example that we combine the ultrasonics treatment together with the photodynamic therapy, because the ultrasonics removal of the dental calculus is just a mechanical disruption of this infected material. But in fact, you are not killing the bacteria that is inside this material. And for the patients, these dental pockets here are highly vascularized, and they are good and very inflamed the tissue. So this area is entrance port for the bacteria for systemic infection. So for patients that have heart valves or chronic kidney disease, there is already a direct correlation of periodontal bacteria to heart infection or kidney disease. So also this prototype in this case was already developed together with a company from the beginning, and this is under clinical trial in 3 different dental schools. This is another project. It was a master project by of Ana Paula Silva, and the supervisor Natalia is here, Natalia Inada. And as in the same case of the PTOs, we first check the in vitro response of the PDT, we set the best parameters, but in this case, there was no animal model. We start already like the design of the best equipment to do this type of elimination. And then Ana Paula finalized her master's thesis with 40 patients, 90 patients, see? Much more higher number. And this is just a single example. So this patient had this lesion for 33 years. So the problem here is because either use antifungal orally, or you have to topically place in the nasal. But the oral treatment is hepatotoxic, and it can take like 6 months, because when you have the drug through your digestive system, it's just a small amount of this drug that is going to be delivered to the nail. And the topical application is not also going to have a high amount of this drug. So in this case, for this patient, took 6 sessions of PDT. In this case, we haven't been testing 2 drugs to set the best protocols. So the best results have been achieved using curcumin, which is a nice photosensitizer because it's from a plant extract. So it would be a much less expensive photosensitizer compared to other ones. So this is just after 6 sessions of PDT, and there is no clinical appearance of these infections anymore. And so besides all these types of experiments and having this protocol for applications in PDT, of course, we also try to understand better all the effects and all the response. So we also use this very nice model to see the vascular effect. This is a chorioallantoic membrane model that we can check the vascular response. So we injected the photosensitizer and we illuminate and taking pictures after this illumination, and we can see and we can even quantify the vascular destruction as a function of time after the illumination period. So the idea is to try to, because when you are with a tumor tissue, you cannot easily separate the response, the vascular response from the cellular response. So in the experiment, in the laboratory, you can try to do this using those type of models. Also, we have been trying to improve the PDT response, modifying some part of this treatment. So this is one example. We have been trying to also compare the PDT response, especially the necrosis depth that we are inducing, using the comparison between our ultra short pulse laser, pulse laser in this case a femtosecond laser, and continuous waves diode laser, which is the irradiation parameter that we use in the clinics. So when we do the comparison of the same energy dose and the same average irradiance, and also, of course, at the same wavelength, in this case, we could have almost twice the necrosis depth compared to the same average parameters with the diode laser at under continuous wave irradiation. So also we have been trying to correlate and to see if we can check with this indirect measurement with the photo bleaching. We can also see that the amount of the photo bleaching that we can use as a direct measurement of the singlet production is in a much higher rate compared to the continuous wave. And this correlates with the response that we had in this animal model, illuminated the liver of the rats. Now we just start doing some more research, understand what would be a better skin model. In this case, the porcine. It's much better skin model compared to the rat, because this is just the autofluorescence of this little pig here, and the structure of the skin is much more similar compared to the rat. When we have this to understand the human response. So in this case, we have also been trying to improve the aminolevolinic acid penetration through the skin using different type of methodologies. So in this case, we have been using also the microneedles, that is just introducing these micro holes in the skin. And the amount of production of porphyrin that we have been checked is much more homogeneous that when we compare if we don't use any type of curettage or these other micro holes, ways that we can deliver better the aminolevolinic acid. Another concern that we have been trying to investigate is if we use multiple PDT sessions, and how is going to be the healing of this tissue, especially the collagen distribution after those multiple PDT sessions. Because one of the advantages of using PDT is the cosmetic effect, because we have a better resolution compared, for instance, with surgery. And in this case, what we saw in Patricia Ramirez's master thesis, that after 4 PDT sessions, we have already diminished and less organized collagen at the epidermis, at the dermal region of this skin. And so probably we would have some type of limit of these multiple sessions, or at least we should wait for longer instead of using several PDT sessions in a short period of time. But this is still under better understanding we should do. And this is another prototype, and also together with the MM Optics company. And also we have a collaboration of this American company that is called True Bios and Johns Hopkins. So this system is for the cervical intraepithelial neoplasia, and is also a dual system that we have here, the wide field fluorescence imaging and the LED treatment probe. And in this case, we want also to see what will be the PDT response using the aminolevulinic acid to treat those type of lesions. So far, we just had 25 patients treated, and the idea is to have been tested here in Brazil, Mexico, and then United States. And for spectroscopy and optical diagnostics, we have been mainly work with fluorescence, spectroscopy, and imaging. And this is just a prototype that we also assemble here in the lab, so we could do the animal models work and also the clinical trials. So this is one example of these collaborations that I have in a dental school. So we have been checking for the detection of the actin K light, which is a ultraviolet induced lesion mainly affect the lower lip, and also for oral cancer. So we have been seeing very nice results also from people from rice. So in this case, trying to check and see the efficacy of these type of optical detections to improve the resolution of the clinician to have a better understanding of the pathology of the lesion already in the clinical setting. So for the wide field fluorescence, was the first prototype, and this was the prototype that resulted in that the visor system and the dual PDT system. And this is just the diagram showing what's the inside that prototype. So we have a LED source or a laser source, and then this is a diquark mirror that's going to reflect this violet light around 400 nanometers. It's going to shine the tissue, and the fluorescence would come back, and most of the violet light will be reflect. And so we'll also have some other filters here, and we can have the naked eye or a digital camera to record this type of image. So this is just one example showing what's the difference when you look at a lower lip with actin K lights and using these wide field fluorescence imaging. So it's the heterogeneities of this type of lesions is enhanced when we see due to the wide field fluorescence. So in this case, we can also do some type of imaging analysis, and the idea that we have been seeing in those patients that the normal lower lip shows a much more homogeneous fluorescence compared to the actinic K lights patients, and this can even help the physician to set what type from the lower lip he's going to take the punch biopsy to final diagnostics. And using the same system, and this is a work that has been performed by Dr. Ana Gabriela Salvi, which is our dermatologist here that is running the non-melanoma skin cancer program and also those optical diagnostics for skin malignancy and skin lesions. So it's just a comparison between the wide field fluorescence image of an actinic keratosis and a basal cell carcinoma, and the comparison with the white light and the wide field fluorescence imaging. And here the seborrheic keratosis for some of non-experienced physicians can look like a melanoma under the white light, but we can see different fluorescence features comparing those type of lesions. And of course, a melanoma diagnostic has a completely different treatment protocol, and seborrheic keratosis is just a benign, it's not even a lesion, but you don't have to remove this type of tissue lesion. And also, of course, this type of system can be used to check the protoporphyrin information production over the time. Enhancing the protoporphyrin production, we can see a brighter red fluorescence. And the discrimination, of course, all this area here were treated by the aminolevulinic solution. So here we can start to see the selective production of the protoporphyrin in these malignant cells compared to the normal tissue, because we don't see that red fluorescence on this normal skin. And just also an important thing, just before placing the cream, it's really important to have this curettage to remove the keratins, to remove the dead cells that we have at the epidermis surface. And we don't do that, the PP9 production is much lower, because of course you don't have such a better, a nice penetration of the cream if you do a curettage. So those are some other examples of those prototypes that we were able to assemble in our lab. So those are, for instance, intraoral cavity LED system. This is for just placing a cell culture plate here, and we have LED systems here to perform an uniform illumination. This is for treating actinic keratosis on the lower arms and on the hands. This is an LED system for operating room lighting. And so the idea is what Professor Svanberger was mentioning. We can closely talk to our engineering and trying to set what type of optical geometry we want for specific illumination systems. So if you want to have any further information from our group, this is our website. And if you click here at research, you would find the 3 areas: atomic physics, biophotonics, and nanoplasmonics. And you can also find our list of publications and patents. And this website is in English, so I think you can all take a look. Also, we have very nice animations that we just have to explain, especially for people that don't know, for instance, photodynamic therapy. We have these cartoons explaining all the process during this photodynamic therapy treatment. And this is the list of our present people from the Biophotonics Lab. So Drs. Lilian Moriyama and Nathalie Nada are our research specialists, so you have been getting to know them for these past days. Those are our postdoc people, PhD students, master students, and undergrad students. And this is just the list of the people here in Brazil that we have been collaborate. Of course, we do need a strong collaborate to medical doctors, to life science institutes, so we can really bring those technologies to test for in patients and in other animal models. And this is my email, also for you to all have my contact. And I guess one thing that we should have here, just to finalize here, is let's make biophotonics cooler than it's even are right now. I guess this is a cool top for research, and I hope you can all improve your academics and continue your work on biophotonics, and let's get that really stronger and stronger and spread all over the world. So thank you very much, and I'll be glad to. In fact, we're really running late today. Oh, and this is the last announcement that I want to make. Tonight we are going to have the dinner, and this is a tasty Brazilian food. And you have in your map, I don't know which spot it is, but this restaurant is called Vai, and we are going to have dinner at 8 p.m. Thank you very much.